my research tries to use those large facilities like European XFEL that produce X-rays and use those X-rays to look at inside of the protein structure. So proteins are in your body, right? And they're used to do a lot of biological functions like respiration, for example. And the proteins I study are very similar to those that are used for respiration. And we use those very special light sources to look inside of those proteins and see how they work. So the SES instrument is a very special instrument that gives us exactly the parameters that we need. So the high repetition rate, the right energy range, the right sample delivery. So it's unique in the world right now. And this allows us to put those proteins in a very thin jet, so thinner than a strand of hair, and then shoot them with those really high repetition rate X-rays, and then get the data that we need to be able to understand how this protein changes structure and then can perform its function. So cytochrome C is a protein, a heme protein, much like the ones that carry oxygen in your blood. And those proteins can perform different functions depending on its structure. So whether they are like more open or more closed. And what we're trying to understand is one of the processes that lead to the structural change. And then we hope that by understanding this process, we can better understand how this actually happens in nature, in biology. The experiment went great. So we measured for almost 7 full days, 24 hours. So it was really fun. And we battled a bit with like jets and things like this. But after it all, we measured 2 different forms of this protein and we have beautiful data. So it worked really well. We're very happy.